Hello and welcome back again. This is Bushra with you, and we're back again for another lesson. Remember, today is our last section in chapter 3. Congratulations if you come this far with me. So, remember, we're going to study a disease related to the system that we studied in the end of every chapter. And this time, we're talking about AIDS, which is the biggest challenge of the immune system because it's going to attack the immune system itself. And we need to learn how's the body reacting when there is no immune system. So to understand the significance of your immune system, this is the best way to relate to it. If you have no immune system or very weak immune system, this is exactly what AIDS is showing us. Now, some of you asking where you can find my booklet. You can find it in Zhyar photocopy in Old Bahar Karun. All right, let's start. HIV and AIDS. Let's see what is the difference between these two vocabularies. Since I see that some of you are using it as if HIV is the disease too. No. The name of the disease caused by this pathogen, remember, this is a pathogen. The disease name is AIDS. Okay? This is a disease caused by this pathogen. What's this standing for? Acquired immunodeficiency syndrome. Acquired means we got it somewhere because of a reason. You did not have it since birth. That's what it means when you say acquired it. Immunodeficiency means weak immunity. Deficiency means decreased, right? Syndrome is disease. And then you have HIV, human immunodeficiency virus, means the human virus that causes the deficiency in your body. Okay, as you know, AIDS is very feared by everyone and for good reasons because curing it is so hard and is one of the deadliest type of diseases on earth. What is the blow that we take, okay, from this disease? It's because the immune system loses its ability to fight pathogens and cancers. Someone without immunity are prone to get infected with any disease, even the easiest one that like a flu for example might kill the person if they don't have immunity. And cancers are going to emerge and nothing is going to control them if there is no immunity. That's why. So to understand this disease and to understand what is this virus is doing, we need to learn about the cycle of this pathogen. What is HIV doing once it's entering our body? This is called the course of HIV infection. So first thing you need to know, if a virus goes to your body, they need to find a cell, a specific cell that there is a receptor on their surface and can find them because of that receptor. And it's matching to their antigen and helps them to enter the cell. Because viruses are obligatory intracellular parasites. If they don't go inside the cell, they cannot replicate, they cannot do any harm to the body, okay? So the cells that are going to host or let the virus replicate inside are usually because of the receptors on their surfaces. In case of HIV, we have immune cells. Let me change the color. Let's do it this way. You have immune cells like macrophage. This is macrophage. And then you also have lymphocytes, okay? Why do I mention these two? Because these two hold a type of receptor called CD4. This CD4, a virus could recognize it easily. CD4. What is a CD4 receptor? Okay, I highly recommend that you start drawing with me. All right, now this one has CD4. If a HIV comes here, they can easily attach here. That's HIV. Okay, and attach here. However, the problem here is that just attaching to a receptor will not guarantee that the virus is going to go inside. So what do we need here? We need another thing which is called a co-receptor to enter. A co-receptor helps virus to enter the cell. Okay? The HIV virus, when it comes to the body, can only recognize a type of co-receptor called CCR5. They cannot understand the other types. For example, the T lymphocyte has a co-receptor, but it's not CCR5. So this virus cannot enter. Okay, it can bind, but cannot enter. Because a receptor like CD4 is for binding and anchoring itself. But for going inside, we need a co-receptor. HIV at the beginning can only recognize a CCR5 co-receptor. So naturally, it's going to go inside the cell with CCR5 and start replicating and making even more viruses. Okay? So the first cell that is going to host our HIV is the macrophage. Okay? However, after a while, they replicate it. They need to go out. When they go out, they're going with budding process. Budding. They take some of, the outside. This process of emerging, like if you studied in exocytosis, emerging with some of the cell membrane of the cell without killing the macrophage. This is called budding. This is budding process. This is how they are going to replicate and spread around and go to more macrophages and are going to infect those macrophages, and again and again and again they use it again. Okay, so it becomes like a factory for these cells. Let's go back here. See, it says once the HIV enters the bloodstream, that's just going inside your body, HIV can bind to CD4 receptors, which is on the surface of macrophages. This is, for example, showing the virus, and this is the surface of the macrophage. This is macrophage. You can tell that there is CD4, and also another co-receptor that the virus can recognize it, which is CCR5. What is this for? Binding. This is just from binding. What is the associated one for? It's for entering, okay? Now, let's see. It replicates, like we said, it entered the cell. It replicates inside the macrophage. They go out by a process called budding. When they go out with budding, that means they are not going to kill the macrophage. They are going to use it again and again. The budding process is helping the survival of the macrophage, and this is how they continue. They go to more macrophage and more and more and more. The point of change happens. It could be 2 years afterwards, could be 10 years afterwards. But the only time when the cycle change is when there is a mutation. This is the most dangerous part. What do we mean by that? See, during one of these mutations, replications, there will be a change in the sequence of the DNA of the virus as a mistake. But sometimes it might help it. This mutation that happens for an HIV inside a macrophage might give it an ability to find other co-receptors and enter other cells. I will repeat it again. So they are replicating in the macrophage again and again and again, okay? During one of these replications, there will be a mutation that's going to help the virus to infect other types of cells in the body, like T lymphocyte. Remember, T lymphocyte had a receptor, but it was not compatible. Now, the mutated virus, there you go, after mutation we call mutated virus, can infect the T lymphocyte. Let's go back. Let's see, there you go. Now, the first virus could not attack it, but let's change the color, showing that there was a mutated virus. This is a mutated virus, for example. Oh, we did not choose the color. Okay. What is this? A mutated virus. It happens randomly, by the way, the mutation. So that mutated virus can recognize the other co-receptor, The book did not name the co-receptor, but just says other co-receptor. And this is helping it to go inside the lymphocyte. However, the T lymphocyte, is a bit unfortunate because there's no budding process here. They will rupture the cell membrane of this cell in order to come out. So that's how they kill the T lymphocytes. Again, the mutated virus is going inside the cell through the other co-receptor because the mutation helped it. The mutation made it possible. Now they can go into the cell or enter the cell through other co-receptors. Got it? Once they are there, they start replicating, same process. After replication, they need to go out, but they don't do budding. What do? They rupture the cell and burst the T lymphocyte. This is how they kill the lymphocyte. So this one doesn't die. Okay, but this one dies. Basha? Good. There you go. Let's go over it again. So mutated virus can enter helper T cell. The first initial one could not enter. Why? Because it doesn't recognize other co-receptors. Okay? Now, unlike macrophage, T helper cells are destroyed in the process. There is no budding. They will be destroyed. And killing T helper cells is the end of the immune system. Because if you remember, we studied in the previous section that the whole humoral and cellular mediated specific immune response starts when the T helper cells release interleukin 2. If there's no T helper cell, no interleukin 2, and no more defenses, okay? It will cripple your immune system. These two pictures shows, this is budding, by the way, budding process of the virus. This is the virus, okay? All right, what is this? This is a macrophage, shows a macrophage. Okay, and all these red dots are viruses. Okay. Let's check this out. This is a very quick review of what we said. You can take a screenshot and study on this as a very summarized notes. All right? The process is the same. Again, there is the life cycle of HIV. The virus needs to go into the cell. CD4, CCR5 are needed if they are not mutated. Okay, they go into the cell. The DNA of the virus is going to attach to the DNA of the human body cells, and then they start replicating, and more virus coming out. This is a budding process, by the way. The phases of infection. There you go. This is what happens within the cell. What do we see outside? What do we see? If a person have HIV, can you detect it? How many phases are there? What's happening to that person? Let's see. There are 3 phases. Let's not forget, 3 phases. Each phase has its own name, and you need to memorize it by that name too. Okay? Phase 1, it has 2 other names. Phase 1, asymptomatic stage. Asymptomatic means without symptom. A is for negative, no symptom. Infection stage. This is where it starts, the beginning of its infection, and it can also infect other people. All right. Let's see what's happening here. Why it's called a symptomatic stage? Because there's about no symptoms shown on the person. They look very normal. However, what happens inside is that amount of virus increases so much because of replication, okay? And the immune system starts its own attack. The plasma cells make antibody to fight the virus. Means that if you check the blood of a person with HIV after a few weeks, you will clearly see there's a lot of antibody. And that antibody, we could use it in checking, okay, for testing in the lab. Okay, we're going to check for what? The anti-HIV antibody. This is antibody our body creates. It shows positive HIV. That's a very early detection process. HIV infected people may feel well during phase 1, but they also are contagious. They can pass on the infection to others. That's one of the biggest problems. That's actually worst phase for me, because if they don't show any symptoms and then they are going to have contact with the other people, that is going to make sure that the virus is going to um, enter other people's body. But when somebody have symptoms, you make sure that you're limiting your contact. So this phase is actually worst one. See, phase 1 could last up to 10 years. That doesn't mean it's always 10 years, but it could maximize and stretch on and on and on until 10 years, okay? Phase 1, remember, it's asymptomatic. You can infect people. Number of virus high, number of antibody high, okay? An antibody is going to be used for testing. Now, the next stage, when the body starts feeling the problem, okay, the number of the HIV increased, and it's also attacked so many white blood cells that now symptoms are emerging. So what happens? There's still large amount of antibody. It did not go down. Why? Because your B cells and plasma cell especially are doing a great job, and they're still steadily creating this antibody. Now, the number of T cells drops steadily. Steadily is not very fast. It's slowly going down, gradually. Okay? Steadily means that without like stopping. I mean, they're just going and it's gradual, okay? And the virus continues to drop. Remember, again, we had a lot of virus replication here and here too. We had large amount of antibody here as well, here as well. As for T cells did not drop in the first phase, but here it starts dropping. And this is the cause of emerging symptoms. The T cells dropping makes the immunity fails you. And this is why the symptoms are emerging. You see, your lymph gland have a lot of lymphocyte cells in there, so they're going to be swollen because they are trying to control the situation by making more white blood cells, and they start fighting and trying to filtrate all that problems in their lymph as well, which is the HIV. Fatigue and weight loss. The person's body, there's a war inside it. Of course, they will feel tired and fatigued. Fever. Viral infections, mostly they cause a lot of fever. Okay? See, this is one of the things why doctors are asking us, do you have a fever? To see whether we have an infection, especially if it's viral infection. Diarrhea develop or worsen, the body starts to crumble. Some infected people may notice mental changes such as forgetfulness, abnormal thinking. Now the body starts like changing, like we said, and it causes all these symptoms, and even there may be worse symptoms as well. There you go. Look at this. This side is telling us what happens to our T cells, okay? And then this side, the red one, is showing the viral RNA copies. That means it tells us how much RNA do we have and how much virus do we have. You see at the beginning, which is the infection phase, the T cells start dropping and then it starts elevating again. That's how it goes. But the number of the virus increases. Once it's going up for the T cells, the virus starts dropping down again. It's going in a very slow mode, okay? So virus going down because the T cells went up again. The change happens. This two lines shows that we don't know the amount of time. Could be 2 years, 10 years. It doesn't matter. There is a period between phase 1 and phase 2. This is how phase 1 is reacting. At phase 2, you could clearly tell that there is a beginning of worse symptoms. The number of T cell again starts dropping and dropping steadily, like we said, okay? While these dropping, on the opposite end, the number of viruses increase. This is step 2, okay, phase 2. In the phase 3, you can clearly see phase 3 starts when the number of T cells drops to 200 cells per milliliter, okay? This is like one of the worst situations that a person might encounter, having this much or lower number of T cells. This number could never activate your specific immunity and you could not recover from it. That means like it's the end of your immune system. That's the beginning or onset of AIDS, which is the name of our third phase. There you go. We call it onset stage of AIDS. Remember, in phase 1 and phase 2, you were infected with HIV, but you still did not have AIDS because AIDS have conditions. Okay, so AIDS starts here. And it's also called opportunistic infection stage. Why? Let's see. In phase 3, the number of helper T cells drops so low that you no longer can activate your B cells and cytotoxic T cells. Done. Your specific immunity is shutting down. As a consequence, the amount of anti-HIV antibody falls. See, in the other two, the antibody for HIV was very high, okay? It was getting higher, B cells were activated, especially the plasma cells among the B cells. But here it starts dropping. Why? Because there's no longer stimulation to create more. The helper T cells are no longer that, the amount is not sufficient to make it stimulated, okay? HIV levels rise dramatically. Dramatically means like suddenly it becomes very high. There you go, sharp increase. Imagine it took years and years and years to lift it this much, but then in matter of weeks it might leverage and go up this high just because your immunity starts shutting down. The virus continues destroying more helper T cells. You already have a crippled immune system, 200 per milliliter, which is already very bad. But now it's also taking even more down. Now here we call it AIDS. So AIDS, it's the disease when you have less than 200 cells per milliliter in the blood. That's how you diagnose it. You will tell somebody they have AIDS once you see this number, or you will tell them once there might be an opportunistic infection. Okay? What is the opportunistic infection? There are bacteria in our body, there are like some yeast in our body, they're harmless. They're helping us, or they're just like commensalism, they are just living there and benefiting from us and without harming us. Here, they don't cause infections. They are not harmful. But when your immunity is so low, they take that opportunity to infect your body and other parts. Okay? This is why they are called opportunistic, means they take the opportunity when you're weak. One of the opportunistics are like, for example, so there is another thing that we could consider as the onset of AIDS is having rare cancers that don't emerge normally. They're very rare. Only when your immunity is suppressed so much, they start emerging, like Kaposi sarcoma, which is like a type of like cancer that normally people won't get it. But here the immunity is so low that there's nothing to prevent it from emerging. All right? So if somebody says, how do you diagnose AIDS if they have less than 200 cells per milliliter? I mean the T cells. And then if they have cancers that are rare and they don't emerge unless you have like a crippled immunity. And thirdly, opportunistic infections are a great sign of having AIDS. There you go. Opportunistic infections, like we said, illness is caused by pathogens, okay, that produces diseases in people only when they have weak immunity. If those types of pathogen goes to your body, they don't cause any problem. Only people with problems in their immunity. Let's see examples. Pneumonia, for example, pneumocytosis pneumonia, tuberculosis, toxoplasmosis, rare cancers like we talked about. Is it possible to cure HIV? See, there are drug therapy, but they're not curing it entirely. They might suppress the AIDS, okay, and slow the progress of it so that the person can live longer without problem, but that's only if you did not reach the last phases, okay? In the context of biology, why is it claimed that HIV does not directly result in death? That's because HIV is only making sure that the cells are dying of your immune system. But you don't die from that. You die from other types of diseases, taking advantage of your body. Even flu could kill you at that phase because there's nothing to stop it. Okay? So it's not directly involved, but it causes it. Let's see. When we're talking about like something this deadly, we need to know how to prevent ourselves from getting it. So the best way is to check what are the ways that they transmit. How do you get an AIDS? AIDS, you cannot get HIV by shaking somebody's hand or like touching them or like in the air surrounding you or like in the waters and food. You cannot get it there because HIV is very sensitive and they die as long as they're outside of your body. So it's so hard to get it. The first thing you should know, it's only through body fluids, like a body fluid from a person directly going to the other person's body. So we have so many things here we're going to talk about, but the first thing I wanted to focus here in one of the examinations in 2024, I think it was like first sitting, we had a question said AIDS could be transmitted, okay, when the HIV enters the other person's body or HIV infected cells. HIV infected cells means a body cell of a person, okay, but there's HIV inside it. Of course, this is like a bag of HIV. Either way, the person, if their fluid contains either the HIV or the HIV infected cell, they will get the disease. So this was one of the most challenging questions back then. So methods of transmission. The most common method, you all heard of it, is sexual contact. Okay? The sexual contact is the main reason why HIV spreads around. Another one is injecting needles. Okay? Injecting needles or syringes, hypodermic needles, they use it in drug. Okay, when people they're like their life is crumbling and they're like sharing needles because they want to share their drugs, this sharing is what causes HIV because one of them might have it and the other one will get it from the contact of the syringe that is contaminated. Also, something very unfortunate is when a mother have it, it's easily passed down during birth to the baby or even during breastfeeding. Something that is the least possible method is through blood transfusion. It's possible, but it's extremely rare and low. Why? Because every time you donate blood, it's going to get screened and processed, and it's not just going to be given to the person like that. If anybody have HIV, that's like refused immediately. Okay, they don't take blood from just anybody. This is again the methods. And then remember, casual contacts such as shaking hand, air, water, toilet seats, it's not gonna give you HIV. Just stay calm about it. It's not that easy to get it. Vaccines and treatments. Let's see what is the problem with the vaccine if we want it for HIV. Now, we tried to develop and test a lot of HIV vaccines, but the problem is that the HIV is mutating so much that we cannot find it. So the rapid evolution on the gene changing of the virus prevents us to have like effective vaccine. Now, it says as a result, new variants of the virus with slightly different proteins constantly appearing. So the best way is to have like a multi a variant of the vaccine and inject it into the person, which is actually not that highly recommended or like working because it's always updating and updating. So a vaccine against HIV must stimulate the immune system to respond to many variants, like we said. Okay, there's still a chance that you get one that's not in the vaccine. What about drug treatment? Chemicals or like drugs, any types, okay? Now, it should be taken in combination of three, all right? Why? Because the virus is highly resistant. You give it that drug, it gets resistant. You give it another one, they might, if there's two, is going to resist both of them too. But it's less likely to resist all three at the same time. So the doctors are applying three. However, there is two problems with this drug therapy is that they might need to administer this drug like 50 times a day, which is so much. You can only take as many when, yeah. So this is hard. And another thing is that if you take this many, it's going to be very, very expensive. And usually when the people get HIV, it's because they're already in poor neighborhood. They are like using drugs. Their life is, it's not how they want. And this is why HIV prevention is a bit like hard. This is it for HIV. I hope that you study the last part of the these chapters and sections, the section review and end of chapters, because there might be questions coming back there. Don't ignore it. There are all the answers here, and I hope you benefit from today's lesson. See you in the next one. Have a nice day.